If you'll be on page 58 in your notes, we're looking still more at our Punnett squares. And what we did in the last section is we talked about what are called Mendelian traits. Mendelian genetics are going to follow Mendel's 3 rules. So let's go back to 57 and remind ourselves of Mendel's 3 principles. We have the principle of dominance. Remember, dominance means that you have a dominant and a recessive allele. And so when we talk about traits that follow Mendel's rules, these are things like rolling your tongue, having a hitchhiker's thumb, having attached or unattached earlobes. These traits follow Mendel's 3 rules, where there's a dominant version and a recessive version. We're going to be talking today about some traits that don't follow these rules, which is why it's important to review. Mendel's 2nd rule is the rule or principle of segregation, which means to separate. And when we look at the Punnett square, remember the mother, we would write her genotype as heterozygous, big H, little h. But when we set it up in the Punnett square, we segregate or separate because each parent donates half. So that's Mendel's principle of segregation. Then we also talked about Mendel's principle of independent assortment. This is where genes that are on separate chromosomes will be inherited separately. So this might be the gene for eye color, and this might be the gene for hair color. They're on separate chromosomes, so I get them independently. Those are Mendel's 3 rules: dominant, segregation, independent assortment. But now, we are going to talk today about when Mendel's rules aren't followed. So you do need to make sure that you've been practicing all of the Mendelian practice problems, because what comes next are non-Mendelian. So we want to know that non-Mendelian inheritance do not follow Mendel's principles. And we're going to be looking at 5 total. Today we're just going to look at 3. These traits are not just dominant or recessive, therefore they don't follow the Mendelian genetics rules. These are going to be things like cystic fibrosis, hair texture, and your blood type. So we're going to start with what's called co-dominance. And co-dominance really kind of makes a lot of sense if you know that the word co means together. So what we want to do is we want to circle the word co, the prefix co, and write together and the word equal. They are equally expressed. So in co-dominance, now in dominant recessive, you have dominant and recessive. If you have the dominant, that's what you show. But in co-dominance, we have these 2 alleles, and they're equally dominant. Neither of them is hidden. They're both showing up. So both versions of the trait show up in the heterozygote. And so if we look at this, 1, 2, 3, which of these is the heterozygote? It's the one with 2 different alleles. This is the heterozygote. And so if this had been dominant recessive, then there'd be one that was dominant. But it's not. They both equally show up. And so there's a reason that we use 2 different letters. And these are 2 different capital letters. And it kind of makes sense that they're both strong. They both show up. So the main example in humans is something called sickle cell anemia. It's a co-dominant disorder where you can have normal-shaped cells and sickle-shaped cells. Now, if you get NN, you have 2 normal alleles. If you have SS, you have 2 sickle alleles. If you have NS, you have some that are normal and some that are sickle. And so this is called co-dominance, because here in the heterozygote, we would have both of them, some normal and some sickle. And so we set this up exactly like we do a Mendelian Non-Mendelian problem. We just have to know that we interpret the genotypes a little differently. So for example, out to the side on the left, what would it look like if we had a normal person with a person who had sickle cell? And so the phenotype here is normal. The genotype is big N, big N. The phenotype is sickle, genotype is SS. And when we fill these in and we bring them together, we get 100% who would be heterozygous, and they have some normal and some sickle. Now in terms of the disease, this person, this heterozygous, they would actually be normal in appearance because they have enough normal cells to give them normal appearance, but they're actually resistant to a disease called malaria. That's because the disease malaria actually hides in your red blood cells, and if you have some sickle, then you can't be infected. So this is one of the examples of non-Mendelian, because there's not a dominant and a recessive, it's co-dominant. The next example is called incomplete dominance, where neither allele is dominant, and they actually blend, or I want to add in the word here, mix. I like the word mix because I see the I in mix, and I see the I in incomplete, and I can match those two together. So a really good example of blended or mixed is actually in 4 o'clock flowers. A red times a white flower will actually give us a pink flower. And so what we do is we have to, again, use genotype alleles. So a capital letter for both alleles, and one of them gets a prime symbol. So my red is going to be big R, My white is going to be R prime, and my pink, which gets 1 from the red parent and 1 from the white parent, is R prime. There's no allele for pink. It's the mixing of the two alleles that gives us the incomplete dominance. In humans, it happens in the genes for curly hair. So big H is curly, H prime is straight, and so your heterozygote is actually wavy hair. And so this is our example here of incomplete dominance. We have one more example that we want to look at today, and it's called multiple alleles. This is where there are more than 2 alleles for each trait. So for example, in blood type, these are the blood phenotypes. But the blood type alleles are IA, IB, and little I. So you can see that there's 3 alleles. However, you only inherit 2 because you still only get 1 from mom and you still only get 1 from dad. So there's 3 forms, IA, IB, and little I, but you only get 1 from mom, 1 from dad. And so when we look at the combinations, you could get IA. Well, that would definitely give you type A. But it turns out that little I, why is it written as a little letter? That's actually because the little I is recessive. So this gets a little complicated because first, we have to think that there's 3 alleles. And then we have to know that if it's written as a little letter, it's recessive. So IA or IA little I both give us the phenotype of type A blood. IAIB gives us the type AB. What does that tell us? tells us that these two are codominant. So one of the main examples of codominance other than sickle cell is actually your blood type. If you are blood type B, it might be because you are homozygous blood type B, or because you're heterozygous blood type B. So you can see how all of these words that we've been learning, genotype, phenotype, dominant, recessive, codominant, homozygous, heterozygous, all these words are really important. When we look at the genotype that has two of the little i's, then we would be recessive, and that's blood type O. Now, just to help you see what it really looks like, the A gives you the protein in one shape. The B gives you the protein in another shape. A and B will have both proteins, and O will have none of the proteins. So lots of new information in this section, and so this is where we're going to stop. There is a summary box back on page 58, so make sure that you complete that summary box. Although I think there's a problem for us to work, so let's see. Nope. This is where we're going to stop. We're going to do sex-linked traits next time. So make sure that you do your summary box, and we'll work some problems together in class.